Hello viewers, in this particular lecture on microbiology, I shall be talking about the different aspects of the dreadful virus, which is the human immunodeficiency virus, causing a disease of acquired immunodeficiency syndrome or AIDS. HIV actually is a virus that belongs to lentivirus. It's a group of virus where there is an external peplomere followed by an inner capsid, and the peplomere is having some projections, protein projections or glycoprotein projections, commonly called GP120. Another unique feature of this particular virus is that it is having two strands of single-stranded RNA, and at the same time there are some enzymes like the integrase, like the reverse transcriptase, etc. Now this particular virus was detected maybe around 1980 or 81 in US. But of course, even before that, this virus was there, but nobody really understood its existence. And it was just considered to be a disease where people can die because of the failure in immunity. So the first positive observation was observed in a gay population who suffered from a disease commonly called the pneumocystis infection, or in better way it is called the pneumocystis carni pneumonia or PCP. This is actually a unicellular fungi pneumocystis which is colonizing in the lung and causing a dreadful pneumonia which is ultimately fatal. So this was the first positive observation, that people are showing this PCP infection and they are definitely gay. Gay people are more affected. Another such observation was there, that the symptoms of people suffering from PCP was similar to that of tuberculosis, as you can see in this particular X-ray photo or X-ray plate. But there was a second major observation, and that was the introduction of skin cancer by a particular virus, which is the Kaposi sarcoma virus. Now look here, this Kaposi sarcoma virus is having also similar structure, having the outer coat, inner capsid and the RNA, and this binding to specific, say, CD98 receptor of the cell membrane and entering inside. And this KSV is actually causing damage of the skin. It is degrading the muscles of the skin and also affecting the lymphocytes. So it is playing with the muscles and the immunity system together. And the outcome is they show typical external skin rashes like allergic symptom. And this particular disease actually at that time was considered as GRID or gay related immunodeficiency, and it is also a fatal disease. Subsequently, it was in 1981, it was declared by CDC that the GRID is nothing but a 4H disease, because they also found out that the disease is coming from Haiti. So it was correlated in this way: Haitian homosexuals, hemophiliacs, and they are causing this pneumonia-like symptom, or they are causing this typical infection in people sharing the same needle. So the fourth H is heroin users. So subsequently this disease was named as AIDS in 1982, and they had this specific set of symptoms.
Sir, I have a question.
Yeah.
When does the symptoms of AIDS become quite prevalent?
You see, just see the symptoms first. It can be fever, it can be pharyngitis, it can be esophagitis, it can be liver disorder, it can be failure of the central nervous system, maybe skin rash. So apparently it is nothing unusual. Initially these symptoms will be appearing maybe 3 to 4 weeks, but actual symptom of HIV will start when the virus has already developed inside the body, and that may take maybe 1 to 2 years or even more. But by the end of third and fourth year, the person will be quite weak. The person will not be producing any immunity cells, and gradually he will succumb. So infection, incubation and expression of the disease symptom really takes time in case of HIV. But coming to the cause of this particular disease of AIDS, which is the virus. This HIV or human immunodeficiency virus was denoted by Jean-Luc Montagnier and Robert Gallo. Two group of people, one working in France, another one working in US. But it is very unfortunate that Montagnier was recognized with the Nobel Prize much later, but Gallo remained as an unsung hero. And what was the observation? Actually, Montagnier discovered a particular virus which he commonly called LAV, or lymph adenopathy associated virus. You can clearly see that this is also having an outer lipid coat with projections. There is an inner capsid, two RNA molecules and those enzymes. Same thing was observed by Gallo, where also he called it as HTLV or human T lymphocyte virus 3, where the same structure was there. So actually they were observing the same virus, and so it was named in 1983 as HIV. Both these two, that is the LAV and the HTLV. But coming to the origin of this particular virus. Now HIV definitely is originating from monkey family. To be specific, maybe the simian, the chimpanzees. So the HIV-1 or HIV-2, they are both zoonotic disease coming from Western Africa or West Central Africa and is spreading in human. And the origin is from chimpanzee, that is the common pan species. And this was the original virus of SIV or simian immunodeficiency virus, which is considered to be the precursor of HIV or HIV-1. And this is the structure of the HIV-1. You can clearly see the projections are there. The wall coat is there, the inner wall is there, and also the capsid and the enzymes. So typically the structure of HIV-1 is there. Subsequently, it was discovered that there is another one which is HIV-2, which is coming from the common sooty mangabey, which is another monkey. And this particular virus is the HIV-2, which is also having the same precursor, SIV, and they are having the same structure. But what additional they have? They also have some MHC protein binding sites. They also have some typical protease or peptidase inside. So maybe they were somewhat more adapted to invade. So that was HIV-2. Now coming to the different ways that this HIV can be transmitted. It can be commonly transmitted through unprotected sex. It can be transmitted through the needles, using the same needle. So it can be transmitted along with blood or tissue fluid. And of course, if the mother is affected, definitely it will get into the child.
So I have a question.
Yeah.
So can HIV be transmitted through saliva?
Technically, simian fluid, saliva, they may contain HIV. But you know, saliva normally is a part of the alimentary canal. So if it is getting into the stomach, definitely that acidic pH will destroy the virus. So even if it contains, but the transmission level is too low. It's not that high. Okay. Now coming to, as I was telling, the HIV-1 and HIV-2, these are the two common species. HIV-1 has high virulence, high infectivity and global prevalence. It is spreading all over the world, and it is commonly arising from the chimpanzee, whereas HIV-2 is having lower virulence. It is only restricted to West Africa, and it is having low infectivity, and it is produced from the sooty mangabey. But a very unique observation is there. Though this particular disease arises from monkey, but the monkey population are not being killed by HIV. So that means what? They have their own resistance to this particular virus, which cannot spread like it does in human. Okay. Now coming to the different steps by which this particular virus will grow in human body. The moment it enters the human body, there is a particular RNA which will be activated and it will be producing the DNA by this enzyme reverse transcriptase. So this is a very important step where the RNA is producing the DNA after the virus is entering the host cell. And this process, as you know, is called reverse transcription. So definitely there is a very important step that if at any means this reverse transcription can be checked, then the virus can be checked. So what happens is, if say for example this is the HIV which is entering the cell, it is activating the reverse transcriptase enzyme and the DNA which is produced is getting integrated into the host DNA. If at this point there is a molecule or an enzyme which is the SAMHD1 which can be blocking the reverse transcriptase, then the cell can be protected. How? It can actually cause apoptosis or cell death, it can actually cause the immune evasion, and it can make the disease latent. And it is very important molecule because it is also preventing those protein particles of that virus to accumulate. Okay.
Sir, I have a question.
Yeah.
So what is SAMHD1 sterile alpha motif?
Yeah. Actually, it is a protein. This protein is enriched with histidine and aspartate. Because it has some alpha motif, it is a sterile alpha motif protein and having enriched histidine.
Okay.
That is having specific binding site to reverse transcriptase enzyme. So thereby SAMHD1 treatment can actually protect potential HIV infected person. Okay.
Yes.
Now coming to the structure of this particular virus in detail. As I was telling you that this virus is unique because it is having multiple layers. There is an outer layer which is the peplomere or the lipid layer or the envelope. Having this projection, there is a stalk of GP41 and the head of GP120. There is an inner capsid, and this inner capsid actually is having the RNA, and this RNA, of course, it is single-stranded double RNA, and there will be enzymes like reverse transcriptase or integrase. But most important is the genome of this particular virus. The genome actually is having a specific genes or specific sites. You can see these particular sites which are P17, P24, P7, P6. Here it is P10, P51, P66, P32, and then there is the GP120, GP41. But broadly the gene is having on both sides long terminal repeats. So this is a very earmark for this particular viral genome. Now coming to this particular gene, when I talk about this gene, there are 9 major genes, 3 major and 6 minor. Now out of these 3 major genes, they are actually this GAG, POL and ENV. But going back, when I compare, this is the typical genomic organization where there will be specifically the structural protein producing genes, the enzyme producing genes and the coat protein. So you can clearly see the GAG is producing structural protein, the POL is producing the different enzymes, and the coat protein is being produced by the ENV genes. Okay? So this particular gene pattern is more or less constant, but it varies in length when I consider separately the HIV1, HIV2. So more or less you can see there is slight variation in the orientation of this GAG, POL and ENV gene. And when I talk about their predecessor from which they have originated, the SIV, it has shown further modification. So what does it indicate? It indicates that the genes were there in their predecessor, but the genes have gradually been modified, evolved. And as it has got evolved, it has got increased infectivity. Clear? Good. Now coming to their function. As I was telling, that the GAG is actually a group of genes that produces the GAG protein, which will determine the core of the retrovirus, retroviral core. The POL, they will actually produce the enzymes. What are those enzymes? Mainly reverse transcriptase, RNase H and integrase. And the ENV produces the coat protein that include the envelope protein, the lipid bilayer, and thereby they will determine specific binding. So they will determine tropism. And coming to the minor protein, the TAT protein, commonly known as P16 and P14, they are transcriptional transactivators for LTR promoter, and they are acting by binding with the TAR RNA. The REF protein or P19 is involved in the off and on movement of the RNA from the nucleus and the cytoplasm. Similarly, the VIF protein or the P23 actually prevents the action of a cell protein that is causing deamination or damage to that virus. So it's a protective protein. Of course, when you attack, you must be protecting yourself from any retaliation from the host. It's obvious. So then we come to three more. The VPR or P14 is arresting the cell division, because the cell division will be arrested in the host cell. G2 and M, that point is stopped. NEF protein or P27 is down-regulating CD4 and MHC class 1 and class 2 molecule. They are also down-regulated. Then that is the reason why the immunity gradually goes down. And the VPU or the P16, they actually help in the release of the new viral particle after the host cell has been destroyed. So this was the structure of the genome. Now let us talk about the different ways by which this virus gradually enters. You see, this is the HIV, and it is binding to specific receptor like the CCR5 or CD4, and they gradually come in with the activation of the V1, V2, V3 loop. They bind, and after this binding gradually they come close, and this is how this GP41 is entering, and the head or GP120 is embedded in the membrane, and thereby this particular movement of this virus close to the membrane has actually put them embedded in the membrane, and gradually this particular folding of the protein or the surface protein, the virus gradually goes in, and as it goes in, the nucleic acid is automatically, the RNA is automatically released inside the cytosol of the host cell. But there is a unique phenomenon. What is that? That it is not really attracted towards a particular receptor. It may be attracted towards many receptors like the CD4, 6, CR4, CCR5, etc. So these particular receptors being attracting the virus, this phenomenon is called tropism.
Sir, I have a question. Sir, what do you mean by HIV tropism?
You see, I have already told you, tropism is a specific movement stimulated by a particular stimulus. So whenever a virus is moving towards a particular receptor, it is actually the tropic movement. And this is the nature of this HIV, that they are not really attracted towards a particular receptor. They may be attracted towards many receptors depending upon their binding nature. So that is how they become dynamic, they become attached to multiple receptors, they get inside the T cells and they cause infection. Okay. Now this is really unique. Apart from this common, the CCR5 or CXCR5, they are also attracted towards the lectin receptor. This D-lectin or DC sign receptors are also very important receptor for HIV, and they also can help in the entry of that virus. Particularly, it becomes successful when there is a tyrosine based motif at the base of the lectin receptor. So that means this tropism is further enriched not only in a particular CD4 or CCR5, but in a separate group of cells having lectin receptor. So that means what? The infectivity increases for HIV. Now, this is a very unique photograph where you can see that the specific CD4 receptor and the CXCR5 or CCR5 present on the membrane, how it is binding to the GP41 and GP120. Normally, at this point, let me tell you, when the virus is entering the cell, if there is a protein which is inhibiting the action, then there is a possibility that this particular virus will not enter. So inhibitory protein may bind this particular receptor and it will prevent the entry of the virus. This is how it can be treated also. But otherwise what happens is this particular virus, which is here it is the GP120, binds with the typical CD4 receptor, and the CD4 receptor now bring it closer. It's very unique. Now this photograph is really unique. You can see this bending of this CD4 receptor will bring in this particular GP120 closer to the receptor of CCR5. And the moment it comes in, now you can see there is a change in the configuration and the infection occurs. So this particular degradation or disintegration of the GP120 actually is helping of the viral RNA to enter, or it is allowing the channel through which the RNA can enter into the host cell. It's a very unique phenomenon. Now this phenomenon of entry is followed by this, I have already told you, reverse transcription. So definitely the reverse transcription means there will be the production of the cDNA, and the cDNA will produce the positive strand DNA, and this positive strand DNA now may have the ability to bind with the host DNA. So this will require another process which is called integrase. So the integrase actually help in the binding of the cDNA with the with the host DNA, and the process is called integration. So what happens is now this particular viral DNA is inside the host DNA, and the viral load increases in the body. So when the protein synthesis of the host cell occurs, you are also producing the viral protein without your knowledge. And then what happens is these proteins or the specific structural proteins produced will be migrating towards the cell membrane, and they will take up the RNA which is newly produced or newly transcribed by the sense DNA produced from the virus, and the complete virus particle will be produced, and they will just come out by evagination. Okay, now this is a very unique slide where you can see that the entry is being affected by the ENV, followed by the reverse transcription with the reverse transcriptase enzyme, RNase and the VIF. Then already this particular viral RNA is inside the nucleus, or the DNA is inside the nucleus, and the DNA is the sense DNA. So it will be adopted by the host DNA. So this is integration brought about by integrase and VPR. And then this host DNA will actually show transcription, producing the viral RNA, and that will be mediated by the TAT and the REV. And this particular viral RNA and the protein produced by those RNA will now assemble with the help of the GAG, POL, NEF and VPU, and this is how the bud is formed, and the budding is giving out the viral particles. So this is a unique life cycle of HIV. Another very important point which I should mention is that the virus is showing modification, and of course it can, because this RNA gets integrated inside the host genome. So the host DNA also is getting mixed. So thereby they can produce different strains. And not only that, you can understand that one is RNA, sense RNA, then there is the sense DNA produced, so they can also mingle. So they will produce innumerable number of viral genomes. And not only that, there may be the another types of viruses attacking at the same time. When the HIV is there, the MLV is the very common one, it will also produce heterodimer. So at every point of time what you can see is the modification of the viral particle due to integration of foreign DNA or foreign RNA into the viral capsid. So more or less this is what is happening, that this particular provirus which had the integrated RNA or integrated DNA will produce the transcription, and then the translation will produce the GAG-POL protein, then there will be assembly and the finished product is given out. This phenomenon of course requires some precursor. So early expressed gene do produce some specific precursor molecules, like the GAG precursor PR55 is one of them. And this will actually target specific areas of the membrane, and this membrane area will show multimerization, and thereby as it multimerizes it will produce the bud, it will bulge out. So the casing is now clear, and now the RNA has got the simple role just to go in and get fitted with it. And the moment it is fitted, there is a unique completion of this multimerization, and as if the membrane has given it out like a bud. So without any stress, this particular phenomenon will take place. The complete machinery is being provided by the ER and the Golgi. You can see these intermediate molecules are all being produced in the Golgi and ER. So it is utilizing your host machinery. Even before killing it, it is coming out of your host body to invade another cell. This is the beauty or dangerous attitude of this particular virus particle. So in other words, this is how it is occurring. The viral particle coming in, getting fused, releasing the RNA, producing the DNA by reverse transcription, and the DNA gets into the DNA of the host cell, and integration is followed by, of course, modification of the cell, and later on it is released. But the greatness of this particular disease is that gradually the number of infected cells increase, the immunity goes down, and a time will come when the person will not be answering to any specific disease and will succumb to that infection. So we have come towards the end of this lecture. What we have discussed today is the HIV, how it originated, what are the different types, who are the ancestral viral particle of this HIV, how they have developed, and most importantly, what are the structural features that make HIV an ideal human immunodeficiency virus.